Hello friends, this video on Biomolecules part 28 is brought to you by examfear.com. No more fear from exam. What is monosaccharides? We have seen mono means one, saccharide means sugar. One sugar. So carbohydrates that cannot be hydrolyzed further to give simple units are called monohydrates. We have seen this. And there are 20 such monosaccharides are known to occur in the nature. Glucose, fructose, they are all examples of monosaccharides. We've discussed this, I'll not discuss this more. Reducing sugar, we have seen this. So anything which has aldehyde, ketones, or hemiacetyl group, right? So they will reduce the filling and Thorin solution. So they are called reducing sugar. If you see all the monosaccharides and all the disaccharides except sucrose, we have seen they are reducing sugars. Main functions of carbohydrates in plant. So one starch is there in the plant, that is carbohydrate, that is used as food storage, and the other is cellulose, that is used to build cell wall. So we have discussed this. So if you want to know in more in details, you can watch the video where we have discussed these carbohydrates. If you classify this into monosaccharides and disaccharides, so we have seen ribose is monosaccharide, deoxyribose is also a monosaccharide, maltose was disaccharide, galactose was monosaccharide, fructose was monosaccharide from foods, and lactose was disaccharide. So we have seen this. If you want, you can see the structures in the chapter where we explained this. What is glycosidic linkage? So this is the link formed between two monosaccharide units. For example, there are two monosaccharide units. There is a link formed when there is a loss of water molecule, right? It is called glycosidic linkage through oxygen. So if you see here, this is OH, and this will form a bond with this guy. Water molecule will be lost, and you'll get this out. So this linkage is called glycosidic linkage. The next question is, what is a glycogen? How is it different from starch? See, it's almost same. Starch is for plants and glycogen is for animals, right? So glycogen is a polysaccharide. Starch is also polysaccharide, but glycogen is used in animals and starch in plant, right? So if you see, the starch are two types, amylose and amylopectin. The glycogen structure is almost similar to amylopectin, but it is more branched, right? Glycogen structure is almost similar to amylopectin, but it is more branched. If you see this is starch, this is glycogen. Glycogen is mostly used in animals, this is in plants. So if you see, this glycogen structure is almost similar to amylopectin. This is amylopectin, but this is more branched. The next is, what is the hydrolysis of sucrose? See, sucrose will give what? You take a sucrose, you get alpha D glucose and beta D fructose. This is what we have seen. See, this is my sucrose. It is giving you alpha D glucose and beta D fructose. We have seen this so many times. What is the difference between starch and cellulose? This also we have seen. This is my starch, this is my cellulose. So cellulose is generally in the cell, outermost part. Starch is in the plant. And if you see the bonding, there's a difference. This is always 1,4,1,4 bonding, and they have two different kind of structure. This is amylose, amylopectin, and this is a straight chain. This is a fiber kind of structure actually. This is cellulose and this is starch. We have discussed this. So you can watch the previous videos where we explained this. What happens when D-glucose is treated with HI? So this is interesting. I have a beta D-glucose. I'm treating this with HI. So if I treat with HI, we have seen this. You get a normal hexane, N-hexane. You can put hydrogens. This is N-hexane. If you treat with bromine water, what happens is Since bromine water is aqueous, this forms a straight chain. The cyclic chain breaks and you get a straight chain. So we treat with bromine water. What happens is you get COH, this is get oxidized, CHOH4 remains intact, and the CH2OH remains intact. This is called gluconic acid. When you treat with nitric acid, what happens? Third one. HOH4. This is nitric acid. So what happens is you get saccharic acid. This is also we have seen. This becomes COH. This CHOH4 remains intact, and this also becomes COH. You get saccharic acid. These reactions we have seen. So we had to give the reactions of D-glucose which cannot be explained by its open structure. See, first thing is glucose does not give 2,4-DNP test or shift test. Right? These two tests are for a free aldehyde. So, but it doesn't give, that means the free aldehyde is not there. So with the open structure you can't explain this reaction because open structure talks about the free aldehyde. Right? It talks about free aldehyde. The next is when you create the pentaacetate of glucose, it doesn't react with hydroxylamine. That is also one thing because the pentaacetate of glucose actually exists only in the cyclic form, even in aqueous medium. So that's why it does not react with NH2OH. The second was pentaacetate one. And the third is glucose exists in two forms, two crystalline form. Right? That is alpha and beta. So you see the alpha has a melting point of 419 Kelvin and the beta has a melting point of 423 Kelvin. These two have different melting points. The question is how they have two different melting points, because they have two different structures, they have two different cyclic structures. The open chain structure is not able to explain these three things. What are essentials and non-essential amino acids? The amino acids which are essential are the one which our body cannot synthesize, right? For example, valine, leucine, there are so many we have explained. Non-essentials are the one which our body can create on its own. Glycine, alanine, there are so many. If you want, we can go to the discussion where we explain 20 different kind of amino acids. Common types of secondary structure of protein. So there are two actually we have seen, alpha helix and beta pleated. So we have seen this beta pleated and alpha helix are two common structure of proteins. This is the one, this is alpha helix, this is beta pleated. Correct? What type of bond helps in stabilizing alpha helix structures? If you see this alpha helix structure, there is a hydrogen bond between NH and CO. So this is a hydrogen bond between NH and CO. So this helps in stabilizing the alpha helix, right? Hydrogen bond between NH group and CO of the adjacent turn. This helps in stabilizing the alpha helix structure. If you differentiate between globular and fibrous protein, this is the structure. Globular looks like globe, fibrous looks like fiber, right? So this fiber is water insoluble, this is water soluble, this is globular and this is fibrous, right? This is generally for structures and this is for enzymes, hormones. So we have seen hemoglobin had this globular kind of structure that we have seen. Explain amphoteric behavior of amino acids. See, amino acid forms Zwitter ion in aqueous medium. So this is my amino acid in aqueous medium It forms deuter ions, plus charge on NH3, minus charge on O minus. It acts both as acid and base, and thus is a amphoteric behavior. So amphoteric behavior is something which acts both as acid and base. Effect of denaturation of structure of protein. So as a result of denaturation, what happens is my helix get uncoiled, the globular structure gets unfolded, and 2 and 3 and 4 degree structures are destroyed, but 1 degree is intact. And also enzymes loses its activity. So if you see, because the denaturation, what is happening is 2 degree, 3 degree and 4 degree structure is lost. 1 degree, that is sequence, is retained. And since the enzymes is all about structure, the structure, the 3-4 degree structure is lost, so enzymes will lose its activity. Correct? It is not reversible. It's a one-way process. Once you get it denatured, you can't get it back. Right? So in this denaturation, what happened? The globules gets unfolded, helix get uncoiled. So this globules get unfolded and this helix gets uncoiled. How are vitamins classified, and what is the name the vitamin responsible for coagulation of blood? See, vitamins are classified between the solubility, fat soluble or water soluble. Fat soluble is A, D, E, K, and water soluble is B and C. We have seen that. And for blood coagulation, I think vitamin K is responsible for blood coagulation. The next is, why are vitamin A and C essential to us? So we have seen that vitamin A deficiency will lead to a disease called xerophthalmia, where the cornea gets hardened, also night blindness. Vitamin C will cause scurvy, where the gums get bleeded. Right? So vitamin A source is fish. We have seen this. Carrot is a good source. Butter and milk. Vitamin C, you use citrus fruits. Citrus fruits, amla, and leafy vegetables. What are nucleic acids and give their important functions? So they told nucleic acids are nothing but they are biomolecules found inside the nucleus of the living cell. That's why they are called nucleic acids, and they are polynucleotides. They are nothing but polynucleotides. Why? Because a lot of nucleotides combine to form nucleic acid found inside nucleus of living cell. Correct? And there are two types. One is DNA, one is RNA. And they have different function. DNA is used to store the information, and RNA is to pass the information and to create proteins by helping ribosomes. Difference between nucleoside and nucleotide. So we have seen this. Nucleoside is nothing but my sugar, this is ribose or deoxyribose, and a base. Nucleotide is nothing but you have sugar, that is ribose and deoxyribose, plus base, and also phosphoric acid. This we have seen. And this nucleic acid is nothing but chain of nucleotides. So if you see, this is the one. This is my sugar and base. So these are my nucleoside, sugar, base, and phosphoric acid. These two are my nucleotides. The next is, the two strands in DNA are not identical but complementary. Why? So if you see, these two strands are held together by hydrogen bond. And there is a logic here. This hydrogen bond is between two specific pairs only. See, the cytosine will always pair with thymine. Similarly, adenine will always pair with thymine. So this is a pair. We have seen this kind of, we had this discussion when we talked about DNA. Thank you. Visit examfear.com to watch more videos, attempt free online tests, get free study materials, find tutors and mentors, and much more. Thanks once again.